We finished talking about the translation last week, Thursday, right? So we kind of finished the genetic expression in prokaryotes, and this is the nice little summary of how prokaryotic genetic expression is different from eukaryotic. Okay, so on the left, the red, okay, is eukaryotic. You can see it right here. And on the right, the black is prokaryotic. Okay, so let's take a look what's the difference. So first of all, remember, in prokaryotes, transcription and translation occur almost at the same time. Once mRNA transcript comes out of the complex between the DNA and RNA polymerase, once the RNA appears, it is translated immediately. Remember that picture here? So the mRNA transcript, which is purple, once it comes out of the RNA polymerase, which is beige, the ribosomes start to translate it right away. That makes sense? Now, little correction. I don't remember if I mentioned that. That's not DNA. That's protein, of course. Okay, that's not my fault. That's folks who made this picture. I'm checking if the mic still works. In eukaryotes, as you remember, transcription, the primary transcription, happens in the nucleus. So mRNA has to be transported to the cytoplasm or endoplasmic reticulum first to get translated. Does that make sense? So that's difference number one. In eukaryotes, transcription and translation are separated in time and in space. Right? Second, in prokaryotes, mRNA, after the transcription, bless you, is ready to get translated. It kind of falls in the same idea. It's ready. It does not require any post-transcriptional modification. mRNA is ready to go. In eukaryotes, we have a number of things that happen to mRNA before it becomes ready for translation. It has to be spliced. Remember cutting out the introns, fragments of the gene that do not encode for any protein sequence. Also, there are other modifications, such as adding the poly-A tail, which allows the RNA to leave the nucleus, and adding so-called cap structure. I'm not going. It's a pretty much a fairly large organic molecule. It's actually a nucleotide, guanosine, that is added to the 5' end of mRNA, and it's a signal for the ribosome to start translation. Does that make sense? So in prokaryotes, there is no post-transcriptional modification whatsoever. In eukaryotes, there is. mRNA has to be changed. Another concept that I'm just introducing here, and I will explain, it's pretty simple. The mRNA, the messenger RNA of eukaryotes, is monocistronic. In prokaryotes, polycistronic. What it means? It means that when DNA is transcribed in, say, bacteria, several genes can be transcribed together, forming one mRNA molecule. Does that make sense? So your mRNA molecule may look like this, where this is the gene 1, gene 2, gene 3. And then this whole molecule will be translated, but separate polypeptides are going to be made during translation. In eukaryotes, the mRNA will always contain one gene. You see, monocystronic versus polycystronic. Does that make sense? And of course, the ribosomal structure is different. You may say, well, wait a minute, but RNA polymerase structure is different too. And I wouldn't argue, but we focus on ribosome that much because the molecule is much bigger. Well, it's not even a molecule. Ribosome is huge. And if you can inhibit different parts of the ribosome from functioning, if you can mess with the ribosome, you can mess with the replication, right? With the translation. And as you can see, a huge number of antibacterial drugs target the ribosome. Does that make sense? So you have to know the differences between eukaryotes and prokaryotes. All right? Pretty much, and explain, why there is no simultaneous transcription and translation in eukaryotes. Because you have, first of all, nuclear membrane, transcription is in nucleus, translation is in the cytoplasm. And because mRNA, when it's just transcribed, has to be spliced, has to be capped, capped, has to be polyadinylated, and so on and so forth. Any questions on that different part? Now, when proteins come out of the ribosome, in prokaryotes, for the most part, proteins are ready to perform their function. They're good to go. Proteins that are fresh out of the oven in eukaryotes often have to be post-translationally modified. Okay. Again, post-translational modification, different alterations to the protein, are most characteristic for eukaryotes. It does not mean that they are absolutely absent, 100% absent in prokaryote. Does that make sense? You see, it's not absolutely yes for eukaryotic organisms and absolutely no for prokaryotic. It's mostly eukaryotes, very little in prokaryotes. Does that make sense? It's not that categorical. Now, when we say post-translational modification, what do we mean? You can cut it. The protein is synthesized, and then often it has to be modified by cutting the piece from it. Several chemical groups may be added, groups like acetyl group, alkyl group, methyl group, phosphates, sulfates, fragments of sugars, and so on and so forth. Sometimes even proteins covalently bind to the freshly synthesized protein in the processes like sumoylation or ubiquitination. Now, the role of those proteins that are added to the molecule of new protein is to direct them somewhere. Like, so the protein, the freshly synthesized protein molecule can travel to a certain part of the cell. Of course, it is characteristic for mostly eukaryotes. Does that make sense? Any questions on that? Yes. I don't remember what simulation does. It transports the protein maybe to the vesicles, maybe it directs them for exocytosis. I need to look it up. Remember that simulation is the big deal in the virus infection. Simulation regulates cellular responses to virus infection. That's all I can tell you. Ubiquitination, you're right. Ubiquitination is the funny thing. Cell destroys the... Ubiquitin, possibly. No, it's okay because look. You just forgot. I don't even remember. Okay, so I came across it, and I know that it's post-translational modification, but that's about it because I never ran into it in my life. Ubiquitination, pretty much what ubiquitination is, what Olivia refers to, when protein is faulty, bad, doesn't look right. The ubiquitin molecules, it's rather small proteins, are attached to the faulty protein and they kind of label it for destruction. That make sense? Like you put on the couch, you know, trash. It's pretty much the same. It's a whole entire pathway in the cell. Bless you. Now this is the sort of a summary slide. And we will walk ourselves through it, but this would be a good sort of a graphic study guide for you through the gene expression in eukaryotes. Because if you can walk yourself and explain step by step, or maybe redraw it, you know, with explanations, you would be certain that you know what you're talking about. So here is the prokaryotic gene expression. Okay, you can see that DNA, which encodes with a gene, the DNA. Sometimes genes have certain regulatory sequences. Genes get transcribed into mRNA, and mRNA is translated into proteins. That makes sense? That looks fairly easy. With eukaryotes, it's a little bit different. First of all, genes have multiple exons and introns. So after the transcription, the primary RNA has to be processed, spliced. Introns have to be cut out. That makes sense? And then those processed transcripts will be translated into protein, which then can be further modified via adding different chemical groups. Does that make sense? You don't have to worry about this. It's epigenetic modification, which we don't talk about too much. Does that make sense to you? So try to walk yourself through the scheme. Any questions?
So the top part was prokaryote?
Yeah. Yeah, so let me put it. So, pro. Okay? Does that make sense? Any questions again? Don't hesitate to ask, like, if you have them later. If you have them now. If you have them ever. Now we're going to talk about DNA replication, which, remember when we started to chat about molecular biology of the cell, I told you that there's going to be a lot of abstract concepts. So in my personal opinion, DNA replication is the most abstract concept of them all. Conceptually, it's a very, very simple idea. DNA is copied during each time the cell divides. Okay, now I'm going to ask you a question. The replication, does it have anything to do with gene expression? No. those processes are not connected, actually. When genes are getting expressed, are all genes in the genome get expressed? No. When DNA is replicated, is all DNA replicated? Yes, the entire genome is copied. Does that make sense, that statement? Okay. So that allows the transmission of genetic information from the parental cell to the daughter cell. Okay. The conceptual mechanism is unbelievably simple. And I'm serious. It's really, really simple. So you have double-strand DNA. The DNA gets unzipped, and molecules of DNA polymerase make new strands. Okay? So in this initial double-strand molecule, technically speaking, each strand carries the entire genetic information regarding the organism. Okay? So why two strands? Why not just one? Why have two? I mean, just what's the advantage of having two instead of one? Backup. Yep. Because as we will see, DNA is prone to breaks. You can break the strand. You can actually like clip it. Okay? And the single strand break or mutations in one strand can be fairly easily compensated, easily repaired, because you have second one. That makes sense? Okay. We're going to talk about the replication in prokaryotes. Eukaryotic replication is a little more complicated, but conceptually, again, they are the same. And my own reason, I have a reason why I talk mostly about bacteria. Because I don't remember if I mentioned it to you, I don't want to lose the forest behind the trees. I don't want to talk about the gazillion of replication factors or transcription or translation factors in prokaryotes versus eukaryotes, so that you get completely overwhelmed with that amount of information and will not be able to process the concept, the basic mechanism of the process. So we're going to talk about the basic mechanism of the process, right? Which is same in pro and eukaryotic cells. Okay. Now, one interesting thing, where is the DNA in prokaryotes? How do we call this? Nucleoid, yes. What about eukaryotes? Nucleus. Let's I know mitochondria, chloroplast, but let's forget about those two fellas for a while. So nucleus. The actual chromosomal DNA is in the nucleus, right? So in prokaryotes, replication, where's it going to be? Like in the nucleoid, right? I mean, the DNA, double-stranded DNA in the cytoplasm is okay. That's what it is. That's nucleoid. In eukaryotes, replication, where does it happen? In the nucleus, right? In the nucleus, where DNA is. So all double-strand DNA structures will be restricted to the nucleus. Does that make sense? When double-stranded DNA is in the cytoplasm, something goes wrong for the cell. There are molecules in the cell that detect double-stranded DNA in the cytoplasm. Which other molecules you can think of that are totally abnormal in the cytoplasm? What shouldn't be in the cytoplasm? generally DNA. Double strand or single strand DNA shouldn't be there. Does that make sense? All DNA in eukaryotic cell should be restricted to the nucleus. Okay? What else? Yes? Huh? Go ahead, Mark. Well, we get there. We are getting there, but what other structure, what other molecule, macromolecule, not even a virus, a macromolecule shouldn't be? Huh? Well, it's an organelle, but I'm talking about the molecule. Which RNA? Huh? Good, unspliced RNA shouldn't be there, but the problem is that cellular proteins cannot detect whether RNA was spliced or not. But you get, huh? Ribosomal RNA. No, ribosomal RNA is fine. I mean, ribosomes get degraded and ribosomal RNA kind of floats. Oh, messenger. It can be in a cytoplasm because some ribosomes float freely. You are in the path. Think about the structure. RNA molecule. How many strands? 1. Technically. Can you have double-stranded RNA? Technically. Technically. Can you have two can you have two complementary strands in RNA? Yes, you can. There's no such thing in eukaryotic cells, but you can have it. So, there is double-stranded RNA. We fucked up. That's virus. So, there are viruses with a double-stranded DNA genome, single-stranded DNA genome, bless you, and double-stranded RNA. Okay? And all those structures, when they pop up in the cytoplasm, cell knows something goes south, and it triggers the immune response, which usually leads to the cell death. Does that make sense? Just a little note on the side. I cannot, not say anything about my lovely viruses. Okay? I love them. They're perfect. Okay? Huh? No. No. Never. Never. I don't think so. I think human race will take out itself pretty well. Viruses don't have to get involved in that. Now we're going to review the enzymes of the DNA replication, then we're going to take a break. Now, this bunch of enzymes, I gave you this table kind of for a reference, and when we'll get to the grandeur picture, grandeur scheme of the replication, we will review what they do again. This is if you decide to make flashcards, it's kind of a decent reference. So there are, I'm going to go not in the order they are here, but in the order what they do. So think about this. First you have unzip. You have to unzip DNA, right? So that's the enzyme called helicase. It works like the running thing on a zipper. Unzips. Okay? Then you have to prime the synthesis. You have to label, here we go, we start. The enzyme called primase makes the short RNA molecule, so-called primer, that's complementary to the copy, the DNA strand that's being copied. Because without primer, DNA polymerase 3 cannot start the replication. Now, DNA polymerase 3 is the molecule that copies, makes copies, okay? Now, when the copies are made, you still have that RNA primer dangling in the front of the entire copy. You have to remove it. It's RNA, it's not DNA. You have to remove RNA and replace it with the DNA sequence. DNA polymerase 1 does just that. It removes RNA primer and replaces it with the DNA. Now, in some cases, and we'll discuss those cases, your freshly synthesized DNA has multiple gaps. Not gaps like this wide, where you have missing nucleotides, but when two nucleotides are not connected together, not linked. So in this case, the enzyme called ligase stitches those nucleotides together, okay, like glues. When DNA is unzipped, when strands are separated, remember it all happens usually at the normal temperature. We talk about room temperature 37 maybe, if it's a human body. So those DNA strands, when they are separated, what would they want to do? Come back together, yeah. Well, they don't really want because they have no brain, but they will try to come back and form double-stranded DNA. To avoid this, there are single-stranded binding proteins, okay, SSBP. It's a very common acronym that you can find. So single-strand binding proteins keep those two strands separated. The molecule known as sliding clamp helps DNA polymerase III, the protein that actually makes DNA copies, not to fall off. And finally, think about the DNA as a structure. It's not, when we say linear, it's not really linear. When we say circular chromosome, well, come on, it's not really circular. Okay, it's all like tangled. And you can imagine if it all gets tangled like this, it would be virtually impossible to, like this, I don't know if you can see, okay, it's all get tangled like this little wire, okay, it would be impossible for the replication to proceed. So there's an enzyme called DNA gyrase or topoisomerase II, which untangles the DNA. One of the best examples of tangling, have you ever saw a wire phone? Like that's a new generation. Like the, landline, but not just landline when you walk with a tube, but with a wire, with a thing, like coiled wire, right? Now this thing can tangle. If you saw it, you know it. This thing can make the most sophisticated structures. And trying to straighten it up, good luck with that. So same goes for DNA. So DNA gyrase runs pretty much here before the fork and straightens DNA up. Does that make sense? Now the last enzyme is quite fascinating one, so-called topoisomerase IV. The chromosome in bacteria, which shape does it have? Circular. It's circular. Now if you try to replicate the circular molecule, the original one, the copy, going to get tangled like this, like links of the chain. So topoisomerase IV introduces the breaks that allow you to separate the copies. Okay? That make sense? Well, I believe it doesn't, but we'll get to this. Okay, let's take a break, and when we'll come back, we'll continue just discussing the replication.